Swayam Prabha. Digital India. Educated India.
So, we are still continuing the same generation of diversity. So, in last lecture, we discussed, actually there are 3 points we already discussed, like how the diversity is going to happen. First is variable number of VDJ region, then combination of different light chain and heavy chain, and third is the one of the very interesting and very important reason for diversity is the N and P nucleotide addition. It is also called junctional diversity, okay, of antibody or T cell receptor. Now, there is a fourth and one of the important two reason. One is it is only specific for B lymphocytes, it is not happening in T lymphocyte, and this particular reason is not happening during the development. This mechanism or this phenomena is happening only after the exposure to antigen, and this is happening only in case of B cell, not in case of T cell, and this is called somatic hypermutation. Okay. This is known as somatic hypermutation. So, let me go little slowly at the beginning, because this is also very simple and straightforward thing. What is happening? You see that CDR region or HV region, the hypervariable region are the most important or only important, you can say, which interact with antigen, right, that you remember for different way I show you by showing my hand, and some figures are already. Okay, so, CDR1, CDR2 and CDR3 are more important, and this particular interaction between antigen and antibody is non-covalent, mostly hydrophobic, electrostatic, van der Waals, and it all depends what kind of amino acids are there on the interacting region. Okay. So, if this is the antibody, if this is the antibody binding region, if there are some positively charged amino acids here, and if there are some negatively charged amino acid here, it is very tightly it will bind. But if certain happen, so that there are, suppose there are 4 negative charge here and there are 3 positive charge here, okay, equally placed. So, the interaction between this and 4 here will be much better, right. Just one amino acid change same way can increase the affinity or the interaction power. One amino acid change in that interacting region means whatever the antibody structure, ultimate interaction or the final interaction is happening between the CDR1, CDR2 and CDR3 and antigen epitope part, right. So, one amino acid change in either CDR1, CDR2 or CDR3 can change the affinity and specificity both. Like say there is one glutamic acid converted to aspartic acid, not much will change, because both are acidic, right. So, similarly hydrophobic to hydrophobic, similar substitution will not make much change. Okay. But if it is a complete, so suppose one acidic amino acid is replaced by a basic amino acid, definitely it will be a big change. So, one amino acid change in the binding or interacting site of the antibody is very important. Okay. So, that change can do 3 things. One, that change may not have increase or decrease, or may not change the affinity or avidity or their interaction power between specificity and affinity. So, it remains same. So, change does not make any difference. Change can reduce the affinity or specificity, or that change can increase the affinity or specificity. Okay. So, no change, increase, decrease, and fourth thing, it can completely change the specificity or affinity. That means, one antibody used to bind antigen A, but change happened, it is not binding at all with antigen A. It can find something else much better to bind. Okay. So, this anything can happen, right. So, what happened, if you remember, I mean if you remember in our where we are discussing the primary and secondary immune response, I am coming back to that. Okay. So, in primary and secondary response, what happened, there was initially, so this is the days and this is antibody concentration. Okay. So, initially first 6-7 days there is no change, and then it is gradually increase, stay there, go down, and in secondary response it is like that and like this. I said fourth, I mean what is the difference? This is the primary response, this is primary response, and this part is secondary immune response. So, the primary and secondary immune response, there are few differences I mentioned or you already know by now. What are those? One, less time in the secondary response to react, more amount of antibody is produced in secondary response in comparison to the primary, and third one is it is more specific. Okay. Third one is more specific in secondary response. So, specificity increase, and the other one I think I mentioned, but if not then I am mentioning again and explain later again. In primary response, most or all the antibody is IgM, whether in secondary response it is IgG. Okay. That happened, that is called isotype switching. Okay. But this is in the constant domain region, because the effector function is changing, because constant domain is responsible for effector function. But how? Because once, if you remember the clonal selection hypothesis, okay, clonal selection hypothesis is telling that one B cell having a receptor will not change in its lifetime, or the same B cell will produce the same, I mean whatever the receptor it is expressing at the surface, when it is activated it will produce the same sequence for antibody. So, these two are same. Okay. So, now if some B cell get activated in the primary response, how it is making a different or more specific antibody, because B cell is not changing, right, or new B cell is not coming in the same B cell, how come they are producing. So, I mean when it was analyzed like how the antibody become more specific, what was found, there is a special event happening in germinal center, basically in the lymph node or in the secondary lymphoid organ, when it is interacting and more making more, not in the first time, mostly in the second time. Okay. So, primary response is very low, but gradually in the secondary response it is the rate is very high. The event is somatic hypermutation. Normally what happened? Normally during cell division we do not have that many mutation, otherwise cell will die. I mean every protein will change their sequence and enzyme will lose their activity, we cannot survive, right. So, what is happening? In this particular case, there is a special event called somatic hypermutation happen. Many theories are there. I mean some are experimentally valid, like there is a protein called activation induced cytidine deaminase, okay, AID, that makes this kind of change happening. What is happening? And another thing also there, because during this period, I mean during this cell division, what happened? The mutation rate, normally when cell divide, our cell divide, DNA replication happened. DNA replication is not error free, right, 100% error free, it is not possible, because it is an enzyme, it is doing a reaction, any time it can do mistake, and the rate of mistake is 1 in 10 to the 10. So, rate of mutation, that is normal cell division found, is 1 in 10 to the 10. But in case of B cell replication or proliferation in during infection or in lymph node or in the secondary lymphoid organ, the rate of mutation or the rate of error in DNA replication in 1 in 10 to the 3. So, it is much high. Okay. And what is happening as a result, there are lot of mutation happen in all over the sequence, but particularly here, and you if you know it is fine, otherwise you just go and check. There is a term in genetics called mutational hotspot. There are some part of the DNA is always there which is very prone to mutation. Okay. So, what is happening, and another theory, I mean I do not know, I mean how like it or not, another theory was there is that during this multiplication or replication, okay, this B lymphocyte or T lymphocyte and B lymphocyte particularly, T lymphocyte also there, B lymphocyte proliferate very fast, and if you want to do anything in hurry, you will do some mistake, and that is also a reason. The DNA polymerase, if you are push them to synthesize the DNA faster, there is a chance of mistake is very high or more. Okay. Mostly AID induced. So, what is happening? If you see this figure, if you see this figure, what you see here, this is heavy chain variable region and this is light chain variable region. Primary day 7 primary response. Okay, the B cells are proliferating, they are converting to plasma cell producing antibody. During that time also mutation occur. What is the rate of mutation? You see these are the mutation, this red spot are the mutation. You see very few mutation are here and there. In light chain there is almost no mutation, and heavy chain there are few mutation, but mostly they are present in CDR3. Okay. In day 14 secondary response, you see the number of mutation is increased, and most of the mutations are in this shadow region or CDR region, CDR1, CDR2 and CDR3. Most of the mutations are within this CDR region, and if you go day 21, the tertiary response or further infection, the mutation is much higher. Okay. So, this mutation can be either good or bad, because some mutation always possible that some mutation can introduce the stop codon in between the gene, some mutation can introduce the stop codon in between the gene. So, that B cell will not produce the receptor anymore, right, it is a random. So, what will happen? That B cell will die. If this B cell is produced or get a mutation which reduce the efficiency or the specificity, that will also will be no use. We need better antibody, or our immune system need better antibody, better quality antibody which can bind better quality. So, as a result, I mean if you see with time more and more infection happen, you see the affinity of the antibody towards the antigen is increasing, and that is because this CDR1 is getting mutated. Normally, this hypermutation is not very common or not at all common, it is happening only in B lymphocyte. Fortunately, it is not happening in T lymphocyte. Okay. Why? I will tell, I mean I can't tell you exactly why, but how it is advantageous that somatic hypermutation is not there, that I can tell you, like it is absent in T cell receptor diversity. So, this is advantageous in this way. Here what is happening, B cell is continuously producing, and this mutation accumulation can give either non-productive, like the B cell will die, or less affinity, or equal affinity, or higher affinity. More and more the affinity increase, the lower affinity or low grade interaction containing B cell receptor will just not grow anymore or not multiply anymore. So, it is a kind of selection going to happen, a positive selection kind of thing, that better will only attach to the antibody and will B cell receptor, and B cell receptor proliferation is going to happen. So, that is how affinity maturation is going to happen. So, in this affinity maturation is happening in case of secondary or tertiary immune response, and that is the reason that in many cases vaccine, if you see when we are immunization, that booster dose is given. So, first you gave say for tetanus, this is very common, you know, most of us got cut while playing or while cycling, and if you go to hospital, immediately they will ask what is when did you take the vaccine or when did you take the anti-tetanus injection, and they will, depending on that they will give. Say if you say that it is more than a year ago, they will give number 1 and ask you to come after 6 months or 3 months or 9 months for the second, for the third. What that second, third immunization or the booster is doing? Polio. You see so many times polio vaccination is going on the same kid, because every time if you give a exposure to the immune system of that antigen, this affinity maturation will happen. Better quality B cell receptor will be in the memory. So, next time it can handle much better way at the from very beginning. So, this affinity maturation is very helpful in immune system as well as which is coming, but first infection it is not happening that much. You can see the rate of mutation or the total quality mutation, and it is this mutation is again another important thing. It is happening mostly in the CDR region. It is not that only CDR region, mostly in the CDR region, rest that framework region the amount of mutation is not much. Even if it is there, it is not making any difference. As long as it is maintaining the structure, we do not have any problem, because we need an antibody which will interact strongly with antigen and save us or protect us. So, in summary, there are 4 main processes of antibody diversity. Whatever we have discussed so far, the 4 main processes of antibody diversity. First, multiple different copies of each type of gene segment. Gene segment means now we know V, D, J, this kind of segment that makes up the immunoglobulin V region. So, when we say the antibody variable region, that is combination of Vj in case of light chain and Vdj in case of light chain. So, multiple copies, multiple different copies, that is very good. Multiple different copies of different segment present for immunoglobulin variable region, true for both light and heavy chain. Combinatorial diversity arises through the pairing of different combination of heavy and light chain. We all discussed this, I am just summarizing again. Light chain V region to form an antigen binding site. Junctional diversity introduced at the joints between different gene segment, that also we discussed. Somatic hypermutation induced point mutation into rearranged variable region. Okay. So, these are the 4 main reasons. So, now what are the points of antibody diversity? Careful, it is very clearly written 4 main process of antibody diversity. So far we are talking about lymphocyte receptor diversity. Here we are specifically mentioning antibody diversity. Why? I hope you can understand, because first 3 is true for both B cell and T cell. First 3 is true for both T cell and B cell receptor diversity, but the fourth one, the somatic hypermutation, is only happening in case of B cell, not in case of T cell. Okay. So, summarizing the whole thing, whatever I discussed in diversity, I am just reading once, or you can also read, like immunoglobulin genes are rearranged in the progenitors of antibody producing cells. That means, not while they are maturing, before maturation. Complete genes that encode a variable region are generated by the somatic recombination of separate gene segments. That you know, multiple contiguous V gene segments are present, and each immunoglobulin locus like V1, V2, V3, that is what exactly mean. Rearrangement of V, D and J segment is guided by the flanking DNA sequence, that is the heptamer and nonamer. That also you know, the reaction that recombines V, D and J segment involves both lymphocyte specific, that means RAG1, RAG2 protein, and ubiquitous DNA modifying enzyme like Cu protein, then ligase, then your DNA repair or polymerase, and which will tell the complete the sequence. Okay. then the diversity of the immunoglobulin repeater is generated by 4 main process that we just have discussed, right. So, what are the 4 different process? The multiple inherited gene segments are used in different combination, that is whatever we discussed VDJ, same thing, but that is inherited. Here the special point that we inherited those segments from our parents. Variable addition and subtraction of nucleotide at the junction between gene segment, that is the junctional diversity, right. That is contributed a lot, and that is one of the most interesting discovery in antibody diversity development. And to the third, hypervariable region, okay. And third hypervariable region, why? Because VJ recombination in light chain, if you go back my previous lecture, the light chain, there was one nice slide also, the light chain, this VJ recombination and junction region come into CDR3. Okay. So, CDR3 anyway it is very important, because CDR3 actually contribute to the center of the antigen binding site. Okay. So, if you see that this is the antigen binding site, okay. So, this is CDR1, this is CDR2, 2 ends are CDR1 and CDR2, and the fold in such a way the protein CDR3 comes in the middle. Okay. So, slight change in the CDR3 in the center, slight change in the center actually makes the interaction very strong or can make the interaction very weak. So, one change. So, CDR3 is very important region both for T cell receptor as well as B cell receptor, because CDR1 and 2 basically 2 surroundings, and the center part of the antigen interacting site is CDR3. And in case of light chain, which is very much contributed by this junctional diversity, because this is the VJ recombination site, right, which is already figure is shown. So, now after discussing all this part from B cell receptor, let us discuss about the T cell receptor gene arrangement. Okay, they are very similar with the B cell receptor according to the mechanism I am talking about. The T cell receptor gene segments are arranged in a similar pattern to immunoglobulin gene segments and rearranged by the same enzymes. So, life is very simple and straightforward. Once you understand that part, you do not have to study much, you just see few figures and understand what is happening. That I will also do the same thing to you too. Okay. So, here is the alpha chain locus of the T cell receptor. You see all V1, V2, V3 and the numbers are different, that we already discussed. And in alpha chain they have only V and J, they have more number of J, same heptamer, nonamer, same RAG1, RAG2, same TDT, everything is same. Okay. So, there is no point of discussing the same thing again for T cell receptor. You just whatever knowledge you have, you just put it here. In beta chain of the T cell receptor, I hope you remember the T cell receptor is composed of one alpha and one beta chain. It is just looking like, I will look like very much similar to Fab. Okay. I am just reminding you if you just forget. So, here in beta chain, which is very similar to heavy chain, here again which has variable region, then we have diversity region D and we have J segment. Here also in beta chain first DJ rearranged and then the DJ recombined DJ or recombined with the B segment, same way. Okay. Here is the schematic diagram. If you see that V1, V2, V3 like that, then J, then C, and then they recombine to Vj. This is for the alpha chain which contribute here. You see this red and yellow code. So, this is a very gross, but still you can understand that even the J region is contributing to this antigen interacting site, and very important, this is very important, I will show you. I mean, even you do not know much about the MSc, but I will tell something, hope you will understand and remember when I will discuss the MSc. In beta chain, this V, D and J, same way first DJ recombine and DJ recombine with V, and here you see this J segment does not have much role in the diversity or antigen binding or interaction. Here only D and V is very important. So, red and green use see this, but the overall structure excuse me, overall structure is very important. Okay. So, in T cell receptor, the central part is very important. T cell receptor central part is very important. Why? If you remember correctly, T cell receptor cannot recognize antigen alone. It must be presented by MHC. Okay. So, if you remember, if I draw the MHC, like we see, we have seen like this yellow, it is not very clear, right. Let me change the color. Say MHC is red, okay. MHC is red. So, this is the MHC, say suppose this double MHC 2, and here you have a antigen like this, which is say this antigen is like this. Okay. This is the structure. So, now if a T cell receptor interact, how it will interact? It will interact like this. Okay. So, if you see this structure, so this end part or the receptor, outer part of the receptor, T cell receptor is interacting with the MHC only, and central part is mostly important for interacting with the antigen, which is contributed by CDR3. So, CDR3 is very important, because this central region is because antigen binding or antigen recognition along with MHC, or even in case of B cell, the interaction is mainly happening, and this center part is combined by the combination of both light chain and heavy chain in case of antibody, and in case of T cell receptor both alpha and beta chain. So, the central part is very important. So, CDR3 contributing the central domain or central region of the antigen interaction site. Here hope you understand that why this center is more important with respect to antigen, because we have a certain specific number of MHC. Okay. So, that we will discuss what is that, but further timing you just remember or believe me that we do not have huge number of variety of MHC, we have a limited number of MHC. So, recognition of myself or own MHC cannot be that many. So, there is not much variation in this region. So, only the antigen which is variety is unlimited, almost anything can be antigen, anything can create problem in immune system. So, that should be recognized by T cell, which is definitely should be foreign. So, if this foreign thing, so this central part that way is very important, which is contributed by the CDR3 both alpha and beta chain, and this region is that way is very important, but every other mechanism is very same. Okay. Alpha, beta and gamma delta, they are very much similar. Okay. They all have 12-23 rule, they follow both gamma delta T cell receptor and alpha beta T cell receptor. There is nothing, but their arrangement may be different, but the mechanism or the principle is same. So, if you consider this, this is the summary. I am not going to go through read and waste time in this case. So, this is the number of V segment, average V segment of this case of immunoglobulin, and this some are in case of T cell receptor, all are. So, this is immunoglobulin, this is T cell receptor only for beta and alpha beta T cell receptor. This is the number of gene, this is the variety, like number of V gene pairs, how many possible, this is all possible calculation that I was saying, it is not that easy that we did. So, finally, total number of diversity after junctional diversity, it is 5 times 10 to the 13 in case of immunoglobulin, and in case of alpha beta T cell, because this number is much more, and this is number of junctional diversity is also very high. So, they are 10 to the power 18 here in case of 10 to the 13, here the somatic hypermutation is not calculated. So, this is the number of possible receptor one person can have, among which many are very similar to or against our own protein or a self protein. So, that will be deleted So, those who are surviving finally and circulating in our peripheral blood, that is enough. Okay. So, T cell receptor diversity in the third variable region, that just I discussed in the in my hand sketch. It is the same thing. Here you see the CDR2 and CDR3 is this is the MHC region, this is the MHC region. So, CDR2 and CDR1 alpha, 2 alpha, 2 beta and 1 beta, they are mostly interacting with the MHC region, but the central region, this is the antigen, this is ball and stick model, this space field model, this is the antigen where CDR3 is interacting. This violet is the CDR3 and this is the CDR3. So, CDR3 alpha and CDR3 beta both are interacting with the antigen mostly, and rest 1 and 2 is mostly interacting with the MHC part, right. So, that is how this is in crystal structure and much more detail, which I discussed in my hand drawing. Okay. Gamma delta T cell receptor are also generated by same arrangement. So, they are actually what happened, this is very nice. So, what happened, this V delta segment is present here actually. So, if V alpha and this V alpha and Dj recombine, this part automatically eliminated. So, if there is one alpha there will be no chance of V delta. Am I clear? If not, then see, this is the V alpha region and this is the Dj region. So, if one J and V alpha segment, so V alpha and J alpha recombine here, what happened? The whole V delta segment is going out from the chromosome as forming the loop. So, there is no question. Okay. So, this is just to tell you that V delta is interpersed between the V alpha and J alpha region. Okay. আমি একটু পেছনেরটাতে যাচ্ছি, হ্যাঁ পেছনেরটাতে যাচ্ছি, ওখান থেকে একটু কন্টিনিউ, ওখানে শেষ করবো, এটা একটা অন্য জিনিস চলে আসছে। ওকে। So, this loop formation you already know, because we have seen in case of immunoglobulin, like how this if V1 and J1 recombine, how the loop eliminate. So, this loop will be eliminate for ever. So, if there V alpha join, there will be no question of V delta joining. So, in one, this is one of the very nice explanation. So, why T cell receptor is either alpha beta or gamma delta type. So, if alpha beta T cell receptor, there are many other theory or hypothesis for that, how it is decided, but this is once V alpha is joined, V delta is not possible. So, this is the T cell receptor diversity so far. Okay. Hope you understand, and after that you read the book, I hope you will understand. If there is any question, definitely we can clear it later. Bye for today. Thank you.